Isoleucine, leucine, and valine are the three branched chain amino acids. These protein letters get this name because they have a branching in their side chain or unique part, kind of like a tree branch. And speaking of trees, it turns out that if there are problems breaking down these amino acids, your pee smells like maple syrup. Yep, we're talking about maple syrup urine disease, or MSUD. This is a metabolic disorder that is caused by mutations in the branched chain ketoacid dehydrogenase complex, or BCKDH. Wow, that was a mouthful. And speaking of a mouthful, this complex is gigantic, and it's responsible for breaking down these branched chain amino acids. Mutations in this complex can not only can make it so that you can't break down these amino acids, but they also cause the buildup of these amino acids and various side products and like breakdown intermediates in the blood. These amino acids are also competing with other amino acids in order to enter places like the brain. And so these other amino acids, including important ones like glutamate and tryptophan, these can't get into your brain, and so this can mess up the brain signaling. You can also mess up with like ATP import, as well as like the amount of fluid that gets into the brain. And so it can cause developmental delays as well as other problems if not treated early. And with like special diet and things. Oh, and the maple syrup thing, that's just a side product called Sotolone that builds up if you have too many of those precursors. And so it's not like a functional product or anything that we know of. It's just kind of like a spontaneous product that kind of just forms if you have those precursors floating around, and then it happens to smell like maple syrup. And so this is a common sign of the disease, because this disease often shows up initially in infants, at least in the classic cases, and so parents may recognize that there's like a maple syrup urine smell to the diaper. So this is the idea behind MSUD, or not the idea, but I mean this is what causes MSUD, and this is the deal with MSUD. But there's a lot to talk about in terms of how this complex works normally, why it's so important, some really fundamental things about enzymes and coenzymes, and various cool factors like that, as well as other things to talk about isoleucine. So let's dive in and talk about them.
These are the structures of isoleucine, leucine, and valine. And so you can see you have that branching structure, but that branch is located in different places in the case of isoleucine and leucine. So with leucine, the branch is further away from the backbone, whereas with isoleucine, the branch point is closer to the backbone. This has the consequence of making it so that the isoleucine is going to be more sterically hindered. Basically, there's more bulk, and that's going to make it harder for the backbone of the protein to rotate. It's going to allow for less flexibility in the protein structure in regions where there's an isoleucine. Isoleucine and leucine both have butyl groups, so they have 4 carbons, whereas valine has a propyl group. It only has 3 carbons. So valine's going to be smaller than these two. These two are going to have the same weight, but isoleucine, remember, is going to be more hindered in terms of its movements. Whereas isoleucine and valine, these guys have their bulk closer to the backbone. They're going to be harder to rotate around, and isoleucine is going to be bulkier. All 3 of these have in common that in addition to being branched chains, they have aliphatic side chains. Basically, they have these side chains that are hydrocarbon based, and they are not aromatic. So they don't have like circular resonance stabilized rings, which we'll talk about more in another post. But you can think of them as kind of just having chains of hydrocarbons rather than having like circles that have these special properties, things like benzene. And so if you look at like tryptophan and phenylalanine and stuff, those are aromatic, but here we're talking about aliphatic. So isoleucine and leucine are isomers, but they're structural isomers. So basically the atoms, they have the same number of atoms. They're just connected in different ways. So they have the same numbers of carbons and hydrogens in their side chain, but they're connected differently. And they're not just differently in space. So we talked before about how we can have stereoisomers where you have like the L and the D versions of amino acids, and our bodies use the D version or the L versions. But basically, these are like left-hand, right-handed versions. So non-superimposable mirror images, where basically the atoms are just sticking out in different directions. That's not the type of the isomer we're dealing with here. Here we're dealing with actual structural isomers because the atoms are actually connected differently. We'll get back to the idea of stereoisomers later, however, because it turns out that if you have a lot of isoleucine built up, you can get a stereoisomer of it form called alloisoleucine, which can be characteristic of this maple syrup urine disease. What else these have in common is that they are nonpolar. So basically, polarity refers to the partial separation of charges. You get this if the atoms are sharing electrons unfairly. So much more in other posts, and I don't want to spend too much time on it today. But basically, all of those different atoms, those carbons, those hydrogens, those oxygens, they're bound to get together by sharing pairs of electrons, which are negatively charged. If they don't share fairly, the negative charge is going to be unevenly distributed. So normally that negative charge is going to be canceled out by the positive charge of the protons. But if those electrons are unevenly shared, the protons basically are stuck where the center of the atom is, but those electrons are going to be floating around them and in these like clouds. And if that cloud is shifted more towards one of the atoms, then that atom is going to be partly negative, and then the other atoms are going to be partly positive, and this gives you this thing called polarity. Long story short, water is really polar. And it only lets other things that are polar into its network. Polar charge spread too much, and so we call those things hydrophilic. If those things are nonpolar, if they don't have anything to offer the water, we call them hydrophobic, and the water excludes them. So you find them in the center of the proteins. And if we look at isoleucine and we look at leucine and valine, we see that they're very hydrophobic. And so they're typically going to be found huddled in the inside of proteins where they're excluded from the water. So much more on this in other posts, but you can classify these as hydrophobic. And so just to summarize this, these are going to be aliphatic, nonpolar, hydrophobic. So aliphatic, they have these chains of hydrocarbons. Nonpolar, because carbon and hydrogen share fairly, you're not going to get that separation of charge. Because you don't have separation of charge, you don't have, or full charge, you don't have things that the water wants to hang out with, they're going to be hydrophobic, and you're likely to find them on the inside of proteins. Other things that these have in common have to do with their metabolism, so how we make them and how we break them down. All of them, well, we can't make them. They're considered dietarily essential. So these are essential amino acids, meaning that our bodies can't make them ourselves. We need to eat them, get them pre-made through our diet. But we can break them down. So this is the basic overview of the pathway that we're going to go through. With any of these branched chain amino acids, or really with any amino acids, the first step you need to do is to remove the amino group. In the case of the branched chain amino acids, this is going to be done with the branched chain aminotransferase. It's going to remove this amino group, pass it off to alpha-ketoglutamate or alpha-ketoglutarate to make glutamate, leaving me with an alpha-ketoacid. So this is going to be done by a transaminase, and there are different transaminases that will work with different amino acids. And in this case, we're using the branched chain aminotransferase. It's going to leave you with an alpha-ketoacid. And so an alpha-ketoacid, well, an alpha-keto, basically, keto is where you have a carbon with double bonded to an oxygen with two carbons on either side. So this is going to be a ketone, and this is a keto acid because you still have this carboxylic acid group. So we got rid of the amino group, but we haven't gotten rid of the carboxylic acid group. Instead, that's going to be the function of the branched chain alpha-ketoacid dehydrogenase complex. It's going to remove that carbon dioxide. It's going to remove this carboxyl group as carbon dioxide, and it's going to hook this on to coenzyme A. So coenzyme A is going to be this vitamin derived cofactor that we'll talk about. And it's kind of going to serve as a handle where we can swap this part off from one place to another. And this part is going to be different in the case of these different branched chain amino acids because we're starting with different things. So for all of these, we're removing this amino group, or removing this carboxyl group, and then where you have this carboxyl group, now imagine there you have this CoA. And there, now you're going to have this branch, this big branchy part that you're going to break down in different ways. Because you start with different products, you're going to end up with different products. And the different products you end up with can then have different metabolic fates. And so in the case of the leucine, you get acetyl-CoA and acetoacetate. Now, if you watched yesterday's post, you might recognize that these are going to be products that can be used to form ketone bodies. So we call them ketogenic. These ketone bodies are these like alternative energy sources that can be used. They can be shipped through your bloodstream and taken in by other cells to be used as energy, or they can be, or these acetyl-CoA and acetyl-acetyl-CoA can be used to make lipids. And so these ketone bodies are kind of just a way that these lipid intermediates can be transported through the bloodstream, since the lipids can't be transported through the bloodstream. And so these are going to be water soluble, and then they can be converted back into a product that can enter this tricarboxylic acid cycle we'll talk about, or they can be used to make lipids. And so we call amino acids that can be broken down into acetyl-CoA or acetyl-acetyl-CoA ketogenic. Now, this is in contrast to glucogenic. So glucogenic means glucose making. So these are amino acids that can be used to make the blood sugar glucose. And we classify something as glucogenic if, when you break it down, you get a product that is in this tricarboxylic acid cycle. This is also known as TCA, or the Krebs cycle, or the citric acid cycle. We more commonly think about this in terms of breaking down molecules in order to use them for energy. So it's hard to see it in this when you're looking at it like this, because what happens is you have this acetyl-CoA group enters the pathway, and then this gets these carbons get oxidized, which we'll talk about in a minute. And basically they have their electrons removed, and those electrons can remove, they're transferred on to NAD plus and FAD, which can then be used to cash in those electrons for energy later on. But these can also be used to build up these metabolic precursors, these intermediates that can be siphoned off to do other things. And so one of the end product of this is oxaloacetate. And if you're just wanting to get energy, you just keep the cycling going. But if you siphon off that oxaloacetate, you can use it to make glucose. So if we have something that's in this tricarboxylic acid cycle, we can use it to make glucose. If something is ketogenic, we can make it into acetyl-CoA, but in order to enter the pathway, it has to join up to oxaloacetate, which is what we were making in order to make glucose. So if you already have to have this in here, we don't classify these as ketogenic. So in the case of the leucine, what we end up with is acetyl-CoA and acetoacetate. And so these were these two things that were strictly ketogenic, and so we call leucine ketogenic. With isoleucine, well, with isoleucine, if you think about when you break this down, now you can get an acetyl-CoA, but you're also going to get a succinyl-CoA instead of an acetoacetate. And the succinyl-CoA is going to be a component of that TCA, and so we call this glucogenic, but you are also making acetyl-CoA, so it's also ketogenic, so we say it's both. And in the case of valine, here we're just getting succinyl-CoA. And succinyl-CoA, remember, was one of those glucogenic things, so we call this glucogenic. And so you can, with the branched chain amino acids, you get a sense of, it's good examples of what we classify as glucogenic or ketogenic, and much more on that topic in yesterday's post. But you can see that you're ending up with different end products because you're starting with different products. And so when these get broken down, they're going to give you different things, but those different things are all going to enter similar pathways. And once they're in those pathways, then your body doesn't really care where they came from. Okay, so let's start with looking a little deeper into what's happening in this complex. So we're not going to talk too much about that transdeamination because, well, that happens with all these amino acids because you got to get rid of that amino group in a safe way so that it doesn't build up toxic products. And so This trans-deamination is a way that your body can kind of just generically remove that amino group, not have to worry about it, and then do what they want with the rest of the molecule. And in this case, what we want to do with it is we're going to remove that carboxylic acid group, and we're going to do this in with oxidative decarboxylation, which is a mouthful of a word. And so let's talk a little bit more about oxidation. So basically, all of those different atoms are joined together by sharing pairs of electrons. And we talked about how in a nonpolar molecule, these are going to be unevenly shared. But we can also, when we have uneven sharing, or when we have like full loss or gain of electrons, we can refer to this as oxidation and reduction. So what you remember oil rig, oxidation is loss of electrons or electron density, and reduction is gain of electrons or electron density. So sometimes, like when we're talking about these electron carriers like NAD and FAD, we're going to see actual electrons transferred, like whole electrons. But a lot of the time in biochemistry, what we're actually seeing is we're just seeing kind of like the shifting or the redistribution of the electron density. So when they're sharing electrons, remember, oxygen is really electronegative. So if you have a bond to something like a hydrogen, which isn't very electronegative, and you swap that out for a bond to an oxygen, where the oxygen is really electronegative, now the oxygen is kind of going to be stealing the shared electrons. So it's like that, it's like you're losing electrons when you're just losing like electron density. So often, but not always, oxidation is accompanied by a gain of a bond to oxygen, or by a loss of a bond to hydrogen. So hydrogen is kind of like the opposite of oxygen. It's really bad at hugging electrons. And so it's easy to kind of steal hydrogen's electrons. And so if you form a bond to hydrogen, well then that's like you gain electrons. And so that would be reduction. So reduction is often, but not always, accompanied by a gain of a bond to hydrogen or loss of a bond to oxygen. Whereas oxidation is often, but not always, accompanied by a gain of a bond to oxygen or a loss of a bond to hydrogen. And so when we talk about oxidative decarboxylation, well, here we're talking about decarboxylation, so removing this carboxyl group and doing it oxidatively. So basically, you're going to be gaining a bond to an oxygen, or you're going to be losing electron density. And so also, when you're losing an electron, so oxidation is lost. That lost electron in this case is going to be transferred to NAD plus, and this is going to give you NADH. And this NADH, well, it can then go to the mitochondria, and it can be used to help produce ATP through the electron transport chain. So a lot of catabolisms and breaking down molecules, you don't get that ATP yield right away. Instead, what you get are these electron equivalents, these reducing equivalents. Basically, these electron transporters are going to take those electrons that you transported, take them into the mitochondria, and use them to make ATP by passing. So these electrons get passed off from one molecule to another molecule. Those pass-offs are used to drive the pumping out of protons, which builds up this gradient of protons, which then flows back in and turns this little machine, this tiny little machine in your cells, ATP synthase. It's going to make ATP. So you can ultimately get ATP out of it. We're not going to get like a full ATP from a single NADH. This NADH that you produce through this BCKD isn't really, this energy is not the main output of this. Instead, it's really just breaking it down in this controlled fashion, so that you can then have these common intermediates that you can use to do other things. And so the NAD, you get an NADH, but it's not like not the big of a deal in this case. But then remember that you can put that like acetyl-CoA or the succinyl-CoA into this pathway and get more out of it if you're wanting to get energy, or you can use the parts for other things. But in order to do that, you need to break down those weird looking things into things that are more, your body's more generic type things, I guess, and that you can be using to make other molecules. And so your body can function with these smaller precursors and build them up in various ways. So kind of like breaking down those Lego sets in order to get smaller groups that you can then do things with. And so that's going to be oxidative decarboxylation. And what we're going to be doing is we're going to also, in the process, we're going to be passing it on, hooking it on to coenzyme A. So coenzyme A is this vitamin-derived molecules from like vitamin B5. And you can think of it as kind of like a handle that cells will stick acyl groups onto. And so an acyl group is where you have a carbon double bonded to an oxygen, and that's bound to another carbon containing group. Now, hydrocarbon chains, so we just have like carbons and hydrogens. These can be kind of hard to break, or kind of hard to make or break bonds too. But if you have it attached to something like a sulfur, well, the sulfur is going to be a kind of weak spot. So sulfur, when you have a sulfur, it's kind of like forms weaker bonds than, say, an oxygen or than a carbon. The sulfur is going to have this bigger, more diffuse like electron cloud, and it's harder to make these strong bonds. And so when you see a sulfur, you can kind of think of a vulnerable place. And so if we are to hook up this acyl group onto a CoA, where the linkage is through a thioester. And so an ester is where you have a double bond to an oxygen, and then it's bound to, so that's a carbonyl bound to an oxygen, bound to another carbon. A thioester is going to be the sulfur version of this. Now remember, sulfur forms weaker bonds, so this is going to be more vulnerable to breakage. So if we have a connection through a thioester linkage, we're going to be able to pass off groups from one thing to another. Another thing about sulfur is it's very nucleophilic. So this means that basically a nucleophile is going to have more electrons or electron density than it can comfortably handle. So it's going to seek out a nucleus, so something that has positivity. So nuclei, that's where in the atom that the protons live, the positively charged protons. So nucleophile is going to want to seek out something that's partially or fully positive. In the case of a carbonyl, so remember that was the carbon double bonded to an oxygen, the oxygen is going to be very electrophilic. It's going to, or the oxygen is going to be very nucleophilic. The oxygen is going to be very electronegative. It's going to pull away the oxygen density from that carbon, leaving that carbon partly positive. Now, when you have a partly positive thing, well, this is going to be electrophilic. It's going to be attractive to a nucleophile like that sulfur. And so sulfur is also going to be able to attack a carbonyl carbon. And if that carbonyl carbon is next to a sulfur, well, this is going to be an easier break point. So you can have a sulfur attack this carbon and push this off. And so this is an example of where we see like a substitution reaction. And so we can get past this off through different sulfur containing groups using this CoA as this kind of linkage point where we can pass this group off from one thing to another. And remember that we're calling this thing an acyl group, where we have a carbonyl carbon bound to an R. In the case of our amino acids, this R is going to be one of those, in the case of the amino acids, this R is going to be like this whole thing. It's going to be whatever the rest of the amino acid was, all that branched chain stuff. No matter what it is, you have this acyl group because you have this carbonyl carbon. And then you can be thinking of swapping this carboxyl group off for a CoA, which is going to allow you to kind of pass this off more easily from thing to thing and have this handle that you can use. This group is actually going to get passed off first. It's going to actually get stuck onto the enzyme complex and then get passed on to coenzyme A. So let's talk more about this complex. So it looks something like this. This is a crystal structure from actually 1999. So they solved the structure of part of this, and then they used other structures that have been solved to kind of piece together what they think the complex looks like. So that's why you see these dotted lines. It's where the connections aren't exactly known, or where regions of the protein they don't know the structure of. But I like this picture because it's showing you where different things are happening. So you can see that the E2 core, basically the E2 is going to be where groups can actually link onto through this lipoyl group we'll talk about. But it kind of like swings between like E1 and E3 and stuff. And so you can get, it can hold on to the molecule while it's getting processed by the different subunits. And so in E1, this is where our oxidative decarboxylation is going to happen. So we're losing CO2, we're losing that carboxyl group. And in E2, we have this lipoyl group. Basically, we'll talk about this in a second, but this is going to allow us through a sulfur linkage to link on to that alpha keto acid that we just removed. We removed this that's going to allow us to link on to this group that we just removed the CO2 from. So it's not like floating away. We have it stuck in this E2. And then what we're going to do is we're going to actually pass it off to coenzyme A, and this coenzyme A. So we're going to transfer it from the enzyme to the CoA. Then we have to regenerate the enzyme, however, and that's going to be done by the E3 group. And when you're regenerating the enzyme, this is where we're going to actually generate that, that NADH. Because what happens is that we're going to be reducing this group, and so then we're going to have to oxidize it back. When you oxidize something, you're losing electrons, you're passing it on to this NADH, and then this can be used in order to make energy. But you can see that this is a really big complex, and so we have this term that we can use to describe really big complexes like this. So in this case, what this is it's going to be E1 is going to be a heterotetramer. So this means that you have 4 groups, and hetero different. So you have 4 different protein chains, basically. So when you have a protein can be made up of multiple chains or a single chain. In the case of this protein, it's made up of a lot of different chains. And E1 is going to be a heterotetramer, so you have 4 different things, or at least 4 things that aren't all the same. In this case, there's 2 copies of E1 alpha and 2 copies of E1 beta, and mutations in either of these can be associated with MSUD. This is going to be doing that oxidative decarboxylation, and it's going to be using a thiamine cofactor. So a cofactor is kind of like a helper molecule for an enzyme. In this case, the cofactor is the thiamine pyrophosphate, which is going to help the enzyme do its job. Now, speaking of its job, I haven't really mentioned what an enzyme is, but this is going to be something that is going to be speeding up a reaction, or we call this catalysis. It's going to speed up a reaction without getting used up in the process. And so enzymes can do this in various ways, holding things in the right position, providing a nice environment, holding things in place so they don't float around, as we see. And so enzymes have different mechanisms, but they're speeding up a reaction without getting used up in the process. So you can use them over and over again. And so in the case of this complex, we have the E1, which is going to do the decarboxylation. We have E2, which in addition to holding everything in place, is going to transfer that group that you just decarboxylated onto the acetyl-CoA, or onto the CoA. And then you have E3, which is going to reset the system. And so by resetting the system, it's making this an enzyme, basically. It's making it so that this can be used over and over and over again. And then you have regulatory subunits. So there's a kinase and a phosphatase that are going to help regulate the activity. And we'll talk more about all of these different parts in a minute. But you have these 3 main parts of this BCKDH, and then you have these regulatory parts. But remember, E2 is going to hold things in place and kind of pass things off. So you have the E1, which is going to be removing that carboxyl group. Then it's going to get stuck on E2 temporarily. E2 is going to pass it off to the CoA. That's going to float away, and E3 is going to regenerate everything. And in the process of regenerating everything, it's going to take that electron that you removed, pass it off to NAD+, and this is going to give you NADH that can then go to be used ATP, make ATP and stuff in the electron transport chain. This complex can then be used over and over, and what the leftovers from it can then be converted into other products, where you get this divergence after you go through this branch chain a-ketoacid dehydrogenase complex. You get this kind of divergence with the different branch chain amino acids, and therefore you're ending up with different products. But going back to how this complex works, so E1, as I mentioned, it's going to use this thiamine pyrophosphate, and we'll also see that E2 is going to use this lipoic acid. Now, this lipoic acid, this is actually just like a free-floating form, but in the enzyme, it's actually covalently linked. So it's linked through like a strong bond to the enzyme. And there's actually like a bunch of these lipoic acids. I think it was like 24 or something. Basically, there's a ton of these groups that are linked onto the enzyme. And so this is going to serve as a kind of like handle, and it's kind of floppy. It can go from like one place to another place in the enzyme. So it can transfer this group from place to place without it floating away. And remember sulfurs, these are going to be these places where we can break things. We can break the bond and we can attack things. Now, when you have sulfurs linked together like this is the oxidized state. And when these are broken up, you can like think of as hydrogens being there instead of the sulfurs being connected. Remember, when we have a bond to a hydrogen, this is like we are gaining electron density, because the hydrogen is going to let you keep more of the electrons than the sulfur would. And so when you have this reduced, you have two SHs. Or when you have it bound to something else, like a carbon, that's also going to be a reduction. And so when this is broken open, that would be the reduced form. And then we need to reset it in order to make this oxidized form. So you get it like this, where basically you have it bound to this product after you've removed the carboxyl group. Now you have this stuck on here temporarily. You're going to pass it off to CoA, form this acyl-CoA group. You're leaving this in the reduced form, and it needs to be changed back into this oxidized form. And this is done. When this is done, you pass that electron off to NAD+ to get the NADH, which can then go into the electron transport chain. And now this group, this lipoyl group, is going to be oxidized back into this form that now it can then attack the incoming acyl group to form another acyl-CoA. Now, this oxidative decarboxylation, this is kind of like a committed step. This is going to be really, going backwards on this would be really energetically unfavorable. So you want to make sure that you're only committing to this pathway if you really want to. And so this pathway is going to be highly regulated. And so there's these regulatory subunits that are going to help things happen in a controlled manner. So there's a kinase, BCKDK, and a phosphatase, PPM1K. So a kinase adds a phosphate group. So it's this negatively charged bulky group. And if you imagine packing a suitcase and then adding some big bulky thing into it, it can kind of influence how make the protein kind of like have to restructure itself a little. Like a conformational change, where change in the shape of the protein can also like offer binding sites. So there are various things that phosphorylation can do to a protein. We often think about kinases as activating a protein, but they can also just deactivate a protein. And this is what happens in the case of this. This kinase is going to inactivate the complex. And then if you remove that phosphorylation, now you can reactivate it. And so you can remove phosphorylation with a phosphatase. So a kinase is going to add a phosphate group. The phosphatase is going to remove this phosphate group. In this case, the kinase is going to inactivate the complex, and the phosphatase is going to reactivate the complex. So in this way, you're able to control the activity of this complex. Okay, so that's if all good things go right. But mutations in this complex can cause maple syrup urine disease, this potentially serious problem. And so I say potentially serious because depending on the mutations you have, it can have different consequences. So like how severe the mutations are, as well as where the mutations are. So if you have a mutation, say, in E3, this is the thing that's going to be resetting the system. And this is also shared by other dehydrogenase complexes. So the pyruvate dehydrogenase complex and the alpha-ketoglutarate dehydrogenase complex. So if you have a problem with this, you're going to have a problem breaking down other molecules as well, not just the branched chain amino acids. If you have a problem with one of these, then you're just going to have a problem with branched chain amino acids. And I say but it can actually be a really, really big deal. And so it can be a really, really big deal because not only are these amino acids a key source of energy and breakdown products, but they're also going to be, if they're built up, they can cause problems. So the characteristic thing about this MSUD is that maple syrup cell, and that's also the way that it's discovered, is through the smell. And the smell comes from Sotolone, which is kind of like this spontaneous product that can form if you have a lot of those branched chain amino acids building up. So you can get the buildup of branched chain amino acids as well as the alpha ketoacids and various intermediates upstream of wherever the problem was in the processing pathway. And so Sotolone, there's not like an enzyme or anything that makes it. It's just kind of like a spontaneous product, as far as I could tell, but it's going to give it the smell. This is characteristic. Another characteristic thing is the high quantity of allo-isoleucine. So when we talked about leucine and isoleucine, we were talking about them being structural isomers. And so this is when we talk about a structural isomer, they're actually connected differently. Whereas when we talk about a stereoisomer, they're just kind of oriented differently in space. And so I talk about this a lot more in my post on alanine. But you can think of like a right and a left hand. They kind of, things are oriented differently in space. And so they might look like mirror images, but they're not. This is an example of what we call an anti, or they might look like mirror images, but they're not superimposable. So they're not exactly the same. They're oriented differently in space. So this is an example of an enantiomer where you have these mirror images, non-superimposable mirror images. There are also things called diastereomers, which are basically where you have things that are oriented differently, but they're not mirror images. You can get these if you have an uneven number of chiral centers. So a chiral center is a carbon that's attached to 4 different things. And those 4 different things can be oriented in different directions, giving you these different stereoisomers. So all the connectivity is the same in terms of what atoms connected to what atoms, but what direction they're sticking out in space can differ. If you only have a single chiral center, which is the case with most amino acids, you only have that single chiral center at that alpha carbon. So the carbon where the side chain is sticking off of. In this case, you can have an L version or a D version. You think of it as kind of like a chain sticking forward or backwards. And in the case of our bodies, we all only use the L version. So for most amino acids, we only have that single chiral center to worry about. In the case of isoleucine, however, well, now we're going to have a second chiral center because we have that branch point. We have that branch point in the chain, and so that it can therefore stick out in different ways. And so we can end up with diastereomers in addition to enantiomers. So in an enantiomer, that's the mirror image. And so all of the things are swapped. But if you only have one chiral center, well, then you only have to swap that one and everything is swapped. But if you have 2 chiral centers, well, if one of them swapped, you're not going to have a mirror image, but you're still going to have a stereoisomer. And so this is the case with alloisoleucine, is you have a stereoisomer in the side chain sticking a different direction. And this alloisoleucine can build up, and this can be measured and therefore be like diagnostic of this disease. So those are kind of the markers that you could see, but what's actually the scary part is what's actually happening or not happening in the body because of these mutations. Turns out that these branched chain amino acids have to compete with other amino acids in order to like get into the brain and in order to get into other places in your body. And so in particular, to get into the brain, they have to compete with other large neutral amino acids, things like tryptophan, phenylalanine, and tyrosine. And tryptophan in particular is going to be really important in your brain. It's going to be needed to make melatonin and serotonin and all these various things that you need. So if you can't get this into the brain, well, that's going to be a really big problem. And if you won't be able to get this into the brain, if you have a lot of competition from leucine in particular, leucine's really, really hoggy of those transporters. It's going to really have a high affinity for those transporters. And so if there's a lot of leucine, it's going to be hogging things. And so these other amino acids can't get in. And so the tryptophan can't get in. You're going to have messed up signaling. You're also going to have problems with like ATP import and export. You're going to have problems with the water balance. So you can get like brain swelling, edema, various problems that can occur. These problems can be prevented if this disease is caught early and they're put on a very special diet, a very restricted diet without having a lot of these branched chain amino acids. But if it's not detected, then you can get these serious problems. And so there are a bunch of different mutations, and you can have different classifications for the disease depending on where these mutations are. And yeah, so these are all caused by mutations in this branched chain keto acid dehydrogenase complex. But because you have so many different subunits, there's a lot of different genes that are involved that you can have different mutations in, and then you can have a lot of different mutations in those genes. Typically, these diseases are going to be autosomal recessive, which basically means you need two faulty versions of the gene in order for there to be a problem. So these disorders are pretty rare. Yeah, but that's the basics. So the basic breakdown for a branched chain amino acid is you're going to remove the amino group. with a branched chain aminotransferase, remove that amino group, and then you're going to send it through the branched chain a-ketoacid dehydrogenase complex. So dehydrogenase, remember, if you have a bond to, if you're losing a bond to a hydrogen, it's kind of like you're losing electron density, because you can think of a hydrogen as kind of being like free electrons. So if you're dehydrogenating something, you're oxidizing it. This is going to be an oxidative decarboxylation. And so you're going to be removing that carboxyl group, and you're kind of going to be swapping off that carboxyl group for a CoA. Now, this CoA is going to have that vulnerable thioester group. It's going to have that that sulfur that is going to be a kind of vulnerable spot. So you can pass that group off from thing to thing, and you can pass it off to these various groups and then modify the chains, break down those chains further while it's still attached to that CoA, and then send it into a various pathway like the TCA, like the citric acid cycle, Krebs cycle, to either get energy or to use its parts to do other things. And so you're going to get different products depending on what you start with. So in the case of isoleucine, you're going to end up with ketone bodies. You're going to end up with acetyl-acetyl-CoA and acetyl-CoA. With isoleucine, you're going to end up with acetyl-CoA and succinyl-CoA. This is going to be ketogenic and glucogenic. If you start with valine, you're going to get succinyl-CoA. This is going to be glucogenic. And if you start with leucine, you're going to get acetyl-acetyl-CoA and acetyl-CoA, which are then going to be ketogenic. And so, yeah, so hope that helps you understand the breakdown of these branched chain amino acids and what happens when the breakdown breaks down.